This week, we will start to discuss large DNA viruses of eukaryotes. Large viruses have genome which ranges from 25 to 1000 kb, and there are diverse group of viruses. The smallest one are adenoviruses, which have linear DNA and they do not have envelope. The DNA is protected by icosahedral capsid, and as their early gene transcribed immediately upon entry, and they push cell to proliferate, to replicate, in order for them to be able to replicate and form new viruses. Largest one, like intermediate size, are herpes viruses, pox viruses, and baculoviruses. And the interesting fact is about pox viruses, because they do replicate in cytoplasm. You know, the DNA viruses replicate in nucleus, however, pox viruses replicate in cytoplasm. And last group are viruses of algae. They are the largest viruses known. You can even see them with light microscope, and they are up to 500 kilobases, and basically they can have size up to 1 to 2 megabytes. The first of these viruses of algae that were discovered were mimiviruses, and mimiviruses were discovered in the water of cooling towers in London, actually, when there was search for the cause of some pulmonary infections. After them, the megaviruses and Pandora viruses were discovered, and they can be found in coastal waters in South America, Australia, and South Africa. They actually resemble bacteria, and they fool algae to eat them. And basically, once they get inside of algae, they infect their cells and start to proliferate, but they are not really pathogenic for humans. Very interesting are Pandora viruses, because their constitutions, their genes encode for proteins which we don't know what they represent. There are no analog in bacteria or eukaryotes. Even so, the viruses of algae are extremely interesting. So we will not discuss them in more details. However, we will start this week with adenoviruses and herpes viruses, and next week we will discuss pox viruses. So let's start with adenoviruses. As I mentioned before, the DNA, the genome is linear double-stranded DNA, which is encapsulated in icosahedral capsid. And within that capsid, there are several proteins which play a very important role. Particularly, you can see the spikes knob fibers which protrude out of the virus, and basically those attach to cellular receptors. In the genome, at the end, there are repeats, terminal repeat sequences. They are inverted repeats, and each 5' end has covalently bound protein here on 5' end and 5' ends. The name adenoviruses comes from Greek word adenos, glands, because they were first time isolated from adenoid tissues. There are 50 human serotypes, and they can actually also infect cattle, mice, birds, and many other animals. They can cause respiratory and enteric infection in humans, sometimes eye and gastrointestinal infections, but they are not really highly pathogenic for human. Um, Adenoviruses can be oncogenic, however, only in animals. They do not cause any cancers in humans. And the infection is outlined here. The cell receptor, firstly, they have to attach to so-called CAR receptor, Coxsackie and adenovirus receptor, which is immunoglobulin-like receptor. And then they need integrin, particularly adenovirus 2 and 5. They need integrin as a secondary receptor, which triggers endocytosis. Once the pH pH decreases, adenovirus are released and transported into nucleus. And they have three groups of gene: early gene, intermediate gene, and late gene. So early genes, we will particularly focus on E1A, E1B, and E3, because those early genes not only have trigger gene expression of the intermediate genes, they also play a very important role in synthesis of proteins. These proteins play important role in pushing cell to proliferate. Intermediate gene usually triggers synthesis of protein, which initiate transcription and DNA replication, and late gene are mostly structural genes. So once DNA is replicated, late gene are expressed. There is viral assembly of the virus, and once virus assemble, it triggers cell lysis, so cell dies. Now, as I mentioned, E1A protein is a kingpin of adenovirus growth cycle. Let's see what happened in normal cells. We discussed this many times that in normal cells, cell needs to receives a signal through growth factor receptor in order to activate proliferation. And what does it do in normal cells? In a resting cell, retinoblastoma RB binds E2F, general transcription factors, and HDAC. HDAC stands for histone deacetylases. So when CDK cycle-independent kinases are activated, they phosphorylate RB, which release E2F, and also HDAC, and hold HDAC. So basically, there is no more deacetylation, because we know genome DNA needs to be acetylated in order histone need to be acetylated to release the condensation of the DNA. Now, what happened with E1A? E1A would bind to the retinoblastoma here and trigger release of the general transcription factor, so cell can proceed to proliferation. In addition, E1A blocks degradation of P53. There is a mistake in the picture, which is in PowerPoint. It said activation. No, it blocks degradation. So what happened in normal cells? P53 is synthesized, but also MDM2 is synthesized, which binds to P53, triggers its ubiquitination and targeted for degradation. However, E1A also, when it releases general transcription factor, it also stimulates transcription and synthesis of P15ARF. And ARF proteins bind to MDM2, so it inhibits it. So basically, MDM2 now cannot ubiquitinate P53, so there is upregulation of P53. However, the virus doesn't want to trigger apoptosis immediately because it needs that cellular machinery to synthesize its own protein and to replicate its own DNA. So E1B protein, which is another early protein, actually suppresses this E1A activated apoptosis by P53. E1B protein, there is two homologs. 19K homologs actually acts as BCL2, which binds backs and prevent caspase activation. So BCL2 is anti-apoptotic protein. And E1B 55K homolog binds to P53 and blocks P53 dependent apoptosis. So basically E1A and E1B enables virus to push cells through proliferation and inhibit apoptosis up to the time that virus has time to replicate and synthesize new viruses. Another protein E3 is so-called DES protein, which is found in infected cells in Golgi and ER, and also in nuclear membrane. and it triggers apoptosis later on. However, the mechanism is not really clear. Another very interesting protein is E4 or 4 protein, which induces cell death in p53 independent mechanisms. So now, adenovirus infection of human cells result in virus replication and cell death, cell lysis. Therefore, the cell cannot be transformed. However, in rodent cells, basically the replication is not very efficient, and those proteins which trigger apoptosis can transform, actually are inhibited, and basically E1A protein can trigger transformation in the cell culture. I repeat again, in human cells, in humans, virus replicate and trigger lysis of the infected cells, so there is no transformation. However, transformation can occur, for example, in rodent cells. Now, there are many therapeutic applications of human adenoviruses. Remember that they don't cause very serious disease in humans, and they can be very readily manipulated for gene expression. So many recombinant vaccine actually use adenovirus as a gene vector. And basically, those vectors use adenovirus, however, is deleted E1A and E1B genes to eliminate any chance of possible interference with human cell replication and apoptosis. So these replication-competent viruses, however, without E1A and E1B gene, are inserted, and basically they can carry, for example, hepatitis B surface antigen, core HIV antigen, and so on, so they can be used as a recombinant vaccine for highly pathogenic viruses such as Hep B and HIV. Also, there is very interesting observation where adenoviruses, particularly viral strain Onyx 015, which is a mutant which does not express E1B, so can be used as a cancer therapy. How it is? Because this strain replicates in cancer cells and lyse them. However, it does not affect healthy cells. And it is because even you remember normally it suppresses apoptosis p53. However, most of cancer cells have a p53 inefficiency. It's not active. So basically, this virus can replicate to high level in the cancer cells and kill them. So that's something which is considered for new cancer therapies.